First, we're going to have a look at this diagram. So glycolysis is a process that can take place. Firstly, it takes place inside cytoplasm. Glycolysis takes place inside the cytoplasm, and it can occur in the presence of oxygen and also in the absence of oxygen. Glycolysis does not, I must say, depends like it does not depends on oxygen. It can take place in outside in absence of oxygen as well. For example, like in yeasts, we don't have we have glycolysis. We break down glucose via glycolysis, and then it goes to anaerobic pathways for ethanol and for production of ethanol, right. And also in human, we produce lactate under anaerobic condition, like when our muscles are exercising a lot, then we produce lactate because we are in a state of oxygen debt. We're not getting enough oxygen, so our metabolic pathways switch to anaerobic metabolism and we start producing lactate, right. And after glycolysis, we have link reaction. So for anaerobic respiration, we go directly to ethanol or lactate, and this gives us energy in the form of NADH. It gives us energy in the form of NADH. I'm sorry, let me just write real quick on this. Yeah. For anaerobic metabolism, after we're done with glycolysis, what is the product of glycolysis? What do we get? We get pyruvate out of glycolysis, right? So this pyruvate can go either into these aerobic pathways in the presence of oxygen, or it can go to these pathways which are in the absence of oxygen. And in this case, pyruvate is converting into ethanol or lactate. But this is not an energy. It is not a high energy yielding process. The energy we get through this anaerobic respiration of pyruvate is very less as compared to the one we're getting out of this whole aerobic pathways, right? So yeah, so after pyruvate goes into glycolysis, after glycolysis, the pyruvate can enter oxygen dependent pathways like the link reactions, Krebs cycles, oxidative phosphorylation. Well, actually, we need oxygen only for this pathway, oxidative phosphorylation. These all are connected to these, right? So let's start with glycolysis. So I have a really good diagram of glycolysis here. So what is the first step of glycolysis? So glycolysis. So what does glycolysis mean? Glyco means glucose, right? And lysis means breakdown. So we're going to break down glycolysis. So remember, we said glucose is a 6-carbon sugar, and then it was going to break down into 2 molecules of pyruvate, which are a 3-carbon compound, right? This is our pyruvate. So in this process of glycolysis, we're going to break down glucose into glycolysis, two molecules of glycolysis. And we're going to generate ATP, which is an energy carrier molecule. This is going to power our cellular needs. Okay, so let's dive into it. Okay, so we begin with glucose here. So we begin with glucose here, 6-carbon compound. We're going to, so what we say is that glycolysis is divided into 2 phases, energy utilizing phase and energy yielding phase. So what I mean by that is that the initial 2 steps of glycolysis, you see, we're going to utilize ATP. We're going to utilize ATP for the first 2 steps of glycolysis, and these are called as priming reaction. We're going to prepare glucose in such a way that it gives us energy or ATP moving forward into this cycle. Okay, so we get glucose first, right? And glucose is converted into fructose. You see, both of these are isomers of each other. Both are 6-carbon. So what happened is here is isomerization. Glucose was converted into fructose. I must say isomerized into fructose. Then what happened is phosphorylation. Oh, also the glucose was phosphorylated. See, it was isomerized and also phosphorylated. It became fructose, and ATP was utilized to add phosphate group to the glucose, and it became fructose phosphate or fructose 6-phosphate. Then another ATP was consumed to make this fructose 6-phosphate into fructose bisphosphate, like 2. So this fructose bisphosphate has 2 phosphate attached to it. This one has only 1 phosphate attached to it. Okay, now after multiple steps, we get 2 molecules of triose phosphate. So this is when erase it. Sorry. Yeah. So what happened here is we get fructose bisphosphate, right? And it is a 6-carbon compound. So we broke it into 3-carbon, 2 molecules of 3-carbon sugar, right? We broke this fructose bisphosphate into 2 molecules of 3-carbon sugar. So what happened here is fructose bisphosphate was broken down into DHAP and G3P. Dihydroxyacetone phosphate and glycerin D-3-phosphate. Both of these are 3-carbon sugar, and you also have to note that both are interconvertible. You can go from DHAP to G3P, and you can also go from G3P to this way DHAP, right? So what happens here onwards is that these 2 molecules of triose phosphate or 3-carbon sugars then will go further, and they will give us ATP. You see, these are the steps where we're getting energy. You see, ATP is being produced in contrast to the steps we earlier mentioned where ATP was being utilized, and now we see ATP is generated. These are the energy generating steps. And how many ATPs are we getting out of here? So remember, I told you we're getting ATPs out of it, and we want to get energy out of glycolysis. So alongside ATP, we're also going to get NADHs, reduced NADH or NADH. This is a reduced form of NAD. Okay, so 2 ATPs and 2 ATPs, 4 ATPs, right? So after all of these reactions process, you don't need to go into the individual intermediates of this at this point in your studies, because this is not it's very complicated for you at this time. And you can just remember that these are many metabolites, and after going through many metabolite intermediates, we reach 2 molecules of pyruvate. But in this like in between that, we're generating ATP, and also we're getting NADHs. So NADH is a hydrogen carrier molecule. We're going to discuss about it just a few moments later. And we generated 4 ATP here, right? So if we do the math here, you see, for every molecule of glucose, we spent 2 ATPs in the energy, in the priming phase, in which ATP were utilized. And how many ATPs did we produce? We produced 4 ATP, right? So 4 ATPs minus 2 ATPs. What is the net ATP production of glucose, of ATP via glycolysis? Net ATP production is plus 2 ATP and how many NADHs? 2 NADHs. 2 NADH. So this will come in handy later when we're going to utilize this in another process, which is electron transport chain. We're going to utilize this in electron transport chain, right? So after this whole process of glycolysis, let me summarize. We got 2 molecules of 3-carbon sugar pyruvate, and we got a net 2 ATP, or you want to say a gross 4 ATP and 2 NADHs, right? Okay, so now next process in line is the link reaction. Link reaction. So why are we calling it a link reaction? Well, it says it in its name. It is linking glycolysis to Krebs cycle. It is just an intermediate step in it, right? And what's mainly going to happen in this process is we take a 3-carbon sugar and then we transform it into a 2-carbon sugar. So what we're doing is in this step is we're removing essentially 1 carbon from this pyruvate, and then it becomes an acetyl-CoA. Also in this step, we're going to produce an NADH. So 2 NADHs from glycolysis, 1 from link reaction. How many NADH we produced? We have produced till now. We got 3 NADHs and 2 ATPs from glycolysis, right? So after this link reaction has taken place, what we're getting here is a 2-carbon acetyl-CoA, which will go directly into Krebs cycle. This is an absolute requirement for Krebs cycle, right? This is a substrate for enzymes of Krebs cycle.